Merhabalar, ben Astrobiyoloji ve Yaşamın Kökeni takımından Ayşenur Saytaş. Yıldız Teknik Üniversitesi Biyoteknoloji ve Genetik Kulübü olarak bu sene 4.'sünü düzenlediğimiz Astrobiyoloji Konferansı'na hoş geldiniz. Hepinizi aramızda görmekten büyük mutluluk duyuyoruz. Şimdi sizlere program akışını özetlemek istiyorum. Konferansımız bugün toplam 4 oturum olmak üzere saat 11'den 16'ya kadar sürecektir. Oturumlar arası 10'ar dakika molalarımızın yanı sıra öğlen 1 saatlik aramız olacaktır. Etkinliğe katılım belgesini almaya hak kazanabilmek için oturum sonlarında paylaşılan ve aralarda geçen link bağlantılarından ulaşabileceğiniz oturuma katıldığınız dair formu doldurmanız gerekiyor. Katılım belgesi alabilmeniz için 9 oturumdan en az 7'sine katılmanız gereklidir. Ayrıca bugün gün sonunda Origins of Life çalışmaları hakkında oynayacağımız Kahoot oyununa katılarak kitap ödülü kazanma şansını yakalayabilirsiniz. Kahoot oyunumuza katılıp YouTube Biogen hesabından alacağınız ödülleri ve çekilişleri takip etmeyi unutmayın. Şimdiden bol şans dileriz.
Esteemed participants, greetings. I myself Salam from the team Origins of Life. Welcome to the Astrobiology Conference organized by the Biotechnology and Genetic Club of Yıldız Technical University. We are pleased to extend a warm welcome to each one of you. I'll present an overview of today's schedule for you. The conference will continue across 4 sessions, spanning from 11 a.m. to 4 p.m. today. In addition to 10-minute breaks between sessions, there will be a 1-hour lunch break. To be eligible for a participation certificate, you must fill out the attendance form, which will be accessible via links shared at the end of the sessions. To receive the certificate, you must attend at least 7 out of 9 sessions. Additionally, by participating in the Kahoot game about the Origins of Life studies at the end of the day, you will have the chance to win a book prize. Don't forget to join our Kahoot game and follow the prizes and raffles on the YouTube Biogen account. We wish you the best of luck in advance.
Hello again, and we would like to start by introducing our team with Beril today. And I will share my screen right now. Can you see our presentation?
Mm-hmm.
Okay. Yes. I'm the co-leader of Team Origins of Life, as you can see, and we are want to introduce our team before starting today. I'm a 4th grade student at Yıldız Technical University, and my department is Molecular Biology and Genetics. I'm currently working at Tissue Culture Laboratory as a volunteer intern, and I'm curious about plant biotechnology and also synthetic biology, except Origins of Life field.
Can you move your cursor from the... Yeah, okay. All right. Greetings, everyone. I am Beyla Akgöl, a 3rd-year bioengineering student at Yıldız Technical University. Currently, I am doing an Erasmus exchange at KU Leuven, and my interests besides Origins of Life and astrobiology are controlled release systems, polymers, and synthetic biology.
Here's our content, our presentation. Now I want to introduce our student club. You know, every kind of university has opportunities for socialization, and they aim to increase student networking. Yıldız Technical University has a lot of student clubs that work for these aims. And our project team in Origins of Life takes on a student club also, which is Biogen. Biogen was established in 2016 under the name Science and Genetics Society within Yıldız Technical University. Since 2017, it has continued its activities under the name Biotechnology and Genetics Club, Biogen. Since our establishment, we have been contributing by training competent and effective young individuals in the field of basic sciences, reflecting them to the industry, technology, and the public. Also, I would like to point out that we have a very diverse student portfolio. Biogen is a club that continues to produce activities in the field of science, although there are a very wide variety of sub-teams that students can take part in. One of these is the project teams. We have a project board consisting of 7 separate teams. These teams are waste management and sustainability, polymer technologies, genetic diseases, bioinformatics, plant biotechnologies, evolution, and team origins of life.
Our project team was founded by our friend Caner Karabaşoğlu approximately 4 years ago, and we operate within the Biotechnology and Genetics Club at Yıldız Technical University, as Ayşenur said. The team consists of a group of scientists, lecturers, and engineering candidates, and our goal is to conduct research on the origin and evolution of life in the universe while examining life in a cosmic environment alongside our teammates. As a team, we hold weekly journal club meetings on the topics astrobiology, the origins of life, prebiotic chemistry, and protocells. And we invite experts in the field to join our discussions and host them in our sessions. And additionally, we try to participate in competitions and conferences as much as our field allows. Here you can see last year's leader Simay Mercimek, our founder Caner Karabaşoğlu, and this year's co-leaders, me and Ayşenur. And here you can see our dear teammates who have put their heart and soul into their works.
And now we would like to introduce our team mentor too. For the first time Caner created the team, we didn't actually have a team mentor. But after our astrobiology conference, we met with Dr. Toparlak, and after this conference, Caner and Simay communicated with him and asked about being a mentor for our team. We appreciate him for helping us for more than 2 years, maybe. But unfortunately, he couldn't attend our conference today. So I want to give a brief biography that belongs to him. Dr. Toparlak graduated from METU with a degree in biology and was awarded a Fulbright scholarship. He earned a master's in chemical engineering, and he completed his PhD in chemical biology under the Professor Sheriff Mency and collaborating with Nobel Laureate Professor Jack Szostak at Harvard on probiotic chemistry and the origins of life. After the postdoctoral research at Yale, he joined the Bailey Group at the University of Oxford. And he's working on functional nanopore systems, single molecule chemistry, and catalysis currently. What are we doing with our members and our teams, actually? We are organizing weekly based journal clubs, and in these clubs, we involve presentations and discussions on current articles we read on various topics related to our field, such as RNA world, hydrogen events, protocells, and simulation studies. Actually, the simulation studies belongs to our improve our project techniques under the observation of our mentor, of course. We also have the opportunity to invite renewed researchers in the field of our meetings and listen to their work directly from them. And for example, you can see on the left side, Dr. Timmy Liu. She's working on conservates and protocell structures. And you can see right here, Evren Fash. She's a PhD student at University of Wisconsin, and she's working with Betül Kaçar. And we talked about the molecular overview of the origin and evolution of protein synthesis. And you can see a regular meeting, with our mentor too.
Last year, during the organization of Astrobiology and the Origins of Life conference, we reached out to Dr. Bruce Damer, a leading researcher exploring the origins of life on Earth and its sustainable future in the cosmos. Unfortunately, he was unavailable on the day of the conference and couldn't attend. However, when he came to Istanbul, he mentioned that he would like to have a face-to-face meeting with us, giving us an incredible opportunity to getting to know him. In a face-to-face discussion with Dr. Damer, we not only engaged in thought-provoking conversations, but also had the rare chance to examine remarkable samples firsthand. Holding a stromatolite fossil and experiencing the sense of polycyclic aromatics from the Murray meteorite dust was truly an unforgettable and inspiring moment. Additionally, he delivered an eye-opening presentation that broadened our perspective and deepened our understanding of the origins of life. Special thanks to him.
As Beryl said, we don't have so much opportunity for meeting face-to-face, so we're doing our journal clubs online. And we just met our mentor last year face-to-face, and he came with Dr. Alexey Minikovich and talked about their recent experiments and gave us advices about how can we create our own career in the field of we are interesting. In addition, we had a chance for observing the pirates too. Thanks again for their time and valuable advice.
And last semester, we took on an exciting challenge by applying to NASA SpaceX's 2024 challenges, specifically to the Beyond Sunlight and Aquatic Chemosynthetic World Challenge. This challenge pushed us to explore life beyond Earth-like conditions. Unlike ecosystems on Earth that depend on sunlight, we had to envision an oceanic world where life thrives purely through chemosynthesis. Our goal was to design a complex and diverse ecosystem sustained by chemical energy, expanding the frontiers of astrobiology and deepening our understanding of potential extraterrestrial life. And when we opened the website that we created for this challenge, the first thing that pops up is the planets I myself have modeled for this challenge. When we scroll down, you can see the details of the planets and the chemistry our friends work so hard to make sustainable. And here you can see the layers of Aquaverdis, our planet, that I myself have drawn. Here's the team members that have come to the challenge with us. And here is the information about the volcanic activity on its layer with the drawing I made. And this detailed version is made by my friend Aysel Salam. And the last drawing was made by our friend Esin Üsküplü about brine pools and rocks. If you're interested, you can always check out the website for the details. We cannot hear your voice, your microphone is closed.
Sorry for that. Yeah, right now. We attended a GOGEC conference competition this year too. This competition is more about synthetic biology and even we will make a poster presentation next weekend. So we would like to give details about our project too. First of all, I would like to say that we don't have any background about how we could write a synthetic biology project. We just read so many articles about protocell structures and origins of life studies about on it. And we thought that we could create a cell system, or we could follow a similar path to write this project. And it should be a benefit for humans too, for the requirement of the GOGEC competition. So we started with the first choice, which disease should we try to create or found a treatment. And we decided it can be a gastritis because we can control it with a pH-dependent system. After we did research on this topic, we formed our idea. We aim to develop PLGA-based droplet nanoparticles encapsulating eupatillin. Eupatillin, which is a drug for the treatment of KEGA-positive Helicobacter pylori-induced gastritis. And Helicobacter pylori is a bacteria that causes gastritis too. And we should think to ensure stability, biocompatibility, and acid resistance too. Because of this situation, we thought that we could coat the system with 2000 MPEG MMT, allowing for controlled release of the therapeutic agent. In addition, all of this, we add a characterization part to our methods because we need to think for the biosafety part too. The nanoparticles will be characterized using scanning electron microscopy, FTR, XRD, and NTT assays, offering a novel and efficient therapeutic approach. Thanks again to all the team members who contributed to this competition too.
And as we come to today's main course, as Team Origins of Life, we organize our astrobiology and the Origins of Life conference every year, inviting expert researchers in the field along with our volunteer team members. The conference takes 2 days, and the areas our conference focuses on are astrobiology, the origins of life, astrophysics, prebiotic chemistry, deep sea biology, protocells, and lipid structures. Let's take a moment to appreciate the incredible minds that have contributed to our conference in the previous years. Our past participants include leading experts in astrobiology, planetary science, and space exploration. And to give you a glimpse of our past events, we have compiled links to the previous conferences, and you can check them from Biogen's YouTube account. Also, you can reach the content created for this conference as well as this presentation from Biogen's website under the tab Faaliyetler. Here's our team's contact information, and here is Biogen's contact information. Feel free to reach out to us for any questions or anything else. And as we wrap up, I want to sincerely thank my co-lead Ayşenur, our mentor Duhan Toparlak, and our amazing teammates for their dedication and their hard work. And a big thank you as well to all the participants and watchers for joining us today. Your support and curiosity inspire us to keep exploring, and please continue to follow our work and support us. See you next time, and thank you.
Değerli konuklarımız, bir sonraki oturum için 10 dakika ara verilmiştir. İkinci oturumumuz saat 12'de başlayacaktır. Oturuma katıldığınızı belirtmek için bir sonraki oturumda chat kısmında ve aralarda paylaşacağımız linklerden katılım formlarına ulaşabilirsiniz.
Dear guests, a 10-minute break has been given before the next session. Our second session will begin at 12 a.m. To confirm your attendance, you can access the participation form via links shared in the chat and during the breaks.
Değerli misafirlerimiz, şimdi günün ilk konuşmacısı olan Sayın Dr. Martina Preiner'ı Luca konulu sunumunu yapmak üzere davet ediyoruz.
Dear guests, now I would like to invite today's first speaker, Dr. Preiner, to present her talk on Luca.
Hi, hello, good morning, and thank you so much for having me today. Just a brief notification in the very beginning. I myself am not a Luca expert. I use Luca as a concept, so I'm not a bioinformatician, but a chemist by training. But I will try to do my best to give you some insight into how we use Luca as a concept. So my presentation is titled Organic Cofactors as a Connection Between Minerals and the Last Universal Common Ancestor, short Luca. And I'm excited to share some new results from my lab as well today. So that's also how I want to start. Of course, now I cannot move anything. Can you still hear me?
Yes.
Okay,
yes,
but I cannot present my slides are not moving. I'm very sorry. Let me just check.
When you change your slides, we can see it.
I cannot see my slides. That's great. Moving. So okay, so I'm very sorry. Now you should let me just double check if this kind of works.
Okay.
Yeah, okay. Now I will just, can you see it now?
Yes.
Okay, I'm very sorry. Okay, so first of all, I would like to thank, first of all, my current group. So my research group is quite new. It's like barely 2 years old. And I was lucky enough to have an array of amazing students visiting and coming to our lab, and also on the campus in Marburg, which is in like central Germany. I had amazing collaboration partners and colleagues who helped us setting up and understanding science a bit better each day. So what our group is interested in is how did we get here? So to first single cellular organisms such as methanogens and acetogens from here, and abiotic environments such as rock pores, but you can also imagine anything else that you want to imagine as a geochemical environment. So luckily for us, there is a theoretical concept that is called the Last Universal Common Ancestor, or for short, LUCA, that kind of already bridges a bit the geochemical side and the biochemical side. And when I say theoretical construct, I mean LUCA was basically declared or defined as the common ancestor of both bacteria and archaea. And thus you could use the genetic information of bacteria and archaea to kind of reconstruct a physiology of LUCA that has been done several times. There was just the latest paper on the matter was in 2024 by Edmund Moody et al. Because we are mostly chemists, and in our lab, the take-home message that we got from these phylogenetic reconstructions. So that means you basically compare what do bacteria and archaea have in common, and then kind of try to see what is most conserved in the physiology. It looks something like that. We are really focusing here on the carbon metabolism of LUCA. So that means how does actually carbon get into the last universal common ancestor. And although there are some differences between different studies about LUCA, so looking here, for example, at Weiss et al. From 2016 or Moody et al. From 2024, what they have in common is that they depict the last universal common ancestor as an anaerobic autotroph with a reductive acetyl-CoA pathway, or Wood-Lungdahl pathway, as microbiologists would say. And that means, translated, it was CO2 fixing and hydrogen and transition metal dependent in its carbon metabolism. And this type of carbon metabolism, I will come back to it in more detail in a few slides. It is still employed by acetogens, which are bacteria mostly, and methanogens, which are mostly archaea today, that you will also find on current Earth. So from this physiology, we can make a deduction on where did LUCA live. So we would have to look for a geochemically active environment rich in hydrogen, CO2, and transition metals. And lucky for us, nowadays on Earth, we would find these systems. They're called serpentinizing systems, or for short, water-rock interaction, or like more descriptively, water-rock interaction systems that come in many different shapes and options, so to speak. They can be hot and acidic. They can be cool and alkaline. They're even found outside of Earth. So here's an example of moon meteorite that just in 2022 by Andrew Steele et al. Was looked at more closely to see that there are actually also hints of serpentinization on this moon rock, so to speak. And last but not least, they're also considered to be very ancient. As soon as you had hot water and or basically water and hot rock, you would have something serpentinization-like. The most important part about serpentinization in this case is that it is a geochemical source of electrons, and this is the source of electrons that we need also for the acetyl-CoA pathway in this case. So what is serpentinization? I just wanted to give you a small introduction to that. As I said, it's a water-rock interaction, and they already happened 4 billion years ago, so roughly around the time when LUCA is stated to have existed. So that means water is basically hitting the rock and is basically changing one rock called olivine to another rock, serpentinite, producing hydrogen in the process. So as I already said, hydrogen is a great energy and electron carrier. And if you look at those two rocks, it's a little bit very undescriptive if you're not a geologist. But if you look at it more chemically, what happens inside these rocks is that you have iron 2 that is being oxidized to iron 3, and you will basically put those electrons on the protons of water to produce a continuous flow of hydrogen, which is very important in this case because you have a system that always produces hydrogen freshly. So what also happens in these systems nowadays is that CO2 can be fixed abiotically to, for example, methane, but also acetate and other organic carbon compounds. And we also know that the CO2 concentrations on early Earth were a lot higher, so you would also have more CO2 dissolved in the ocean. So that means we can assume that these type of CO2 fixation processes also happened on an early Earth. So what also happens during serpentinization is that you have as side and main products of the whole process, you would have catalytic minerals being formed continuously as well. I will come back to that also shortly. So the question is now, if you have CO2 fixation with hydrogen with the help of catalytic minerals, you have also a continuous production of organic materials. And our question is, could this reaction between CO2 and hydrogen, thus as it is an entry point to microbial carbon metabolism in LUCA, could this also been the reaction that actually started protometabolism to begin with? So as I promised you, I wanted to describe a little bit more what the physiology of LUCA was when it comes to carbon metabolism. So this is the reductive acetyl-CoA pathway. Just briefly, it exists of two branches. It exists of a methyl branch on the left side here and a carbonyl branch on the right side here. Usually there are, depending on the organism, roughly 9 enzymes and 10 cofactors involved in the whole process. So stepwise, in this case, in the methyl branch, CO2 is being reduced to formate and then to a methyl group, further on to acetyl-CoA, which is basically a central molecule that then basically can go anywhere in metabolism. So as a next CO2 fixation step, we would go to pyruvate, and from pyruvate, we would basically be able to build up the rest of carbon metabolism. And as I said, it's still employed by acetogens and methanogens today in this very way. The only difference being that acetogens and methanogens have different energy conservation ways. So, and if we look at serpentinizing systems on the other side, we would have the same starting materials. We would have CO2 and hydrogen and also catalytic minerals to actually help in the very beginning when we wouldn't have any enzymes. So, as I said, what our group is really looking into it. We have a top-down approach when it comes to LUCA. We have a phylogenetic approach. But what our group is trying to do is to present a bottom-up approach with a CO2 fixation with hydrogen as a Kickstarter reaction for metabolism to actually reach LUCA from the bottom up. So As you can already see, reducing carbon is key for not only for life as it is, but also for life's emergence. So what I did during my PhD a few years back is I looked at mineral surfaces as predecessors. I was not the only one, of course, and there were great results when it came to just looking what certain minerals could do when it comes to CO2 fixation. So the connection that we have between enzymes and mineral surfaces are transition metals. So if you, for example, look at hydrogenases, which are really fascinating enzymes, really, because they can make hydrogen, which is actually quite hard to it doesn't want to react very well, let's put it like that, but if it reacts quite violently. But here you have enzymes that actually can make the energy and electrons accessible to metabolism. And if you look into the center of hydrogenases, you would, depending on the hydrogenase, find transition metals such either iron or nickel iron. And those are also transition metals you would find in serpentinizing systems, but also outside of them. So what we did basically is use a variety of different minerals that you would find in serpentinizing systems, like aruite, a nickel-iron alloy or intermetallic compound, an iron sulfide called brigide, and an iron oxide called magnetite, which you would find in serpentinizing systems, and see if under aqueous conditions and we could do CO2 fixation with hydrogen. And what you can see here, the take-home message here is that the main products and only products that we got from these reactions are formate, methanol, acetate, and pyruvate. And if you now look at the Wood-Lunglar pathway here, you would find exactly these intermediates and products in different forms, of course, but in the same chemistry, let's put it like that, as you would find just with minerals. And what our take-home message from that is that there might be a geochemical pattern in biochemical CO2 fixation. And that's from basically the starting point from what my group is doing today. So as I said, we're trying stepwise to approach LUCA. By the way, not LUCA as a single or a cell or anything, but like a community that lived 4 billion years ago. We're trying to do that in a stepwise process, starting from geochemical processes such as serpentinization, then going into very undirected catalysis on mineral surfaces, and then there is quite a jump between an undirected catalysis to something more directed that I called self-sustaining catalytic networks, and then from a self-sustaining autocatalytic network to actually getting to first protoenzymes and a really where you could already recognize a proto-life is still another quite big jump. So what we're really trying to focus on right now is getting from an undirected mineral surface catalysis to a more directed catalytic network. So just as a small explanation why I'm depicting this here as rock pores, that is kind of our premise, that we are within a serpentinizing system that can show small compartments in the form of rock pores. This is just an example that I sampled a few years back with some colleagues at Rainbow Hydrothermal Vent Field, which is also a serpentinizing system, and why we find rock pores such a nice proxy for prebiotic compartments, because we would have catalytic mineral and dissolved ions, we have convection concentration, there's a lot of great papers by other groups on that, we have constant supply of carbon and hydrogen, we have various conditions that can be linked. We also have the possibility of reduced water activity, which basically means there's also some wet-dry cycling happening inside these rocks. And we have the potential for electrochemical potential and also a long time of geochemical activity. So you would really have a long time to actually build something up. So without further ado, so what are we doing? As I said, we're trying to kind of link undirected mineral catalysis to something more self-sustaining. And we try to use coenzymes or organic cofactors as a missing link to reach that goal. So what are coenzymes or cofactors? Those are molecules that basically assist, you can helper molecules for enzymatic catalysis, but actually what is happening there is that the chemistry of certain enzymes is usually linked to the cofactors. So, for example, redox cofactors giving away electrons and accepting electrons again. So this is just one example. It's a so-called iron hydrogenase, and inside this enzyme, you'd find both a methyl donor and acceptor cofactor and a redox cofactor linked within an enzyme. So we see organic cofactors as possible predecessors of enzymes linked with minerals as predecessors as well. They were already described as a possible missing link between metabolism and genetics by many people already decades ago. But we also see them as a possible missing link between geochemistry and biochemistry, and as being part in a pre-enzymatic catalytic network as well. There has been a lot of great theoretical work by a dear friend and colleague of mine, Joanna Javier, that actually showed how coenzymes could help to sustain a self-sustaining catalytic network without enzymes. So looking back or going back to the Wood-Ljungdahl pathway or reductive acetyl-CoA pathway, there are a lot of coenzymes involved in that. So just I grouped them a bit so it's not too confusing, but you have basically C1 donors and acceptors. They come also in many shapes. You have methanofuran, H4-MPT, H4F, and cobamide. You of course have redox cofactors such as the nicotinamides or flavins. And you also, of course, have C2 donors and acceptors in the form of acetyl-CoA. So what is the idea behind this? So let's say we start from, as I showed before, we start from CO2 fixation on a mineral surface. We get reduced carbon out of it. But in order to increase complexity, cofactors could have helped to also get certain functions of mineral surfaces. So in this case, for example, we have a redox cofactor that could make, instead of having hydrides absorbed to the surface to get a soluble hydride donor. Also, what is worth mentioning here is that if you assume that there was a plethora of potential coenzymes or just biomolecules or proto-biomolecules that could have done a certain task, such as hydride donation and accepting, there might have been a selection between different biomolecules if they could do certain tasks under certain environmental conditions. That is what I'm trying to show here. On the other side, you also would have activated reduced carbon metabolites. So that means you would have a cofactor, a proto-cofactor, whatever you want to call it, that would be able to get you activated reduced carbon and also basically getting the reduced carbon off a mineral surface. So, and as a next step, in order to increase complexity, you would have different cofactors interacting with each other, then thus by energy conservation and also feedback loop getting into a more self-sustaining network. So just to give you an example, if you look at the reductive acetyl-CoA pathway and you zoom in a little bit, so we really look into the methyl branch and what coenzymes we would look at there. There are, as I already showed, there are 3 different ones that can be methyl donors and acceptors. And the one that we focused on first was tetrahydrofolate, or for short H4F, which is a C1 donor and acceptor in acetogens, so bacteria. So what did we do there? So this is basically a zoom in on what the methylation route in acetogens is when you really look into the methyl branch of the Wood-Ljungdahl pathway. So the first step is a formylation. So that means you go from H4F to a formulated H4F. Then you have a condensation reaction. That means you get to 5.10 methanol H4F. And then you have 2 reduction steps until you get to a methyl group that then in a last step together with a carbon monoxide gets into to form acetyl-CoA in the end. So a very important step here. So we were looking at an abiotic methylation route of H4F. This is mainly the work of a PhD student of mine, Giuseppe Perosh, who also had help from Patja, who was a summer intern in our lab last year. So what they did is basically start from formate to basically use formate and just try to get as far as possible along the methylation route here. So there was already very quite old literature on the formulation of H4F with concentrated formate under 60 degrees, 3 hours, and naturally very low pHs because of the formate. And so this we knew could happen at least under very concentrated conditions. The question is, as soon as you increase the pH a little bit too much or you get into a more neutral area, you would always have a back reaction. So that is something that we had to consider when thinking about if we could actually also do the reduction steps in one pot to actually get from the methanol H4F to the methyl H4F. So we did this in the very beginning. So you basically can buy this molecule and we just try to reduce it and had quite a low yield, but at least we knew that it would work. So what they did next is basically start from an aqueous solution of formate, so 60 millimolar formate roughly, and just did a one-pot reaction, also 60 degrees, 5 bar of hydrogen as a reductant, and nickel iron powder to actually catalyze the hydrogenation. And after 16 hours at low pH, so it always stayed below 7, we got quite a good yield of almost 13% of this methyl H4F, which is really, really great because it's the first time that someone could show that you actually can make an abiotic activated methyl donor. Abiotically, a methyl, a molecule that can actually donate a methyl group. So we know it works if we start from formate, but if you maybe also remember a few slides back, I showed you that we can produce formate from CO2 fixation with hydrogen. So naturally, we wanted to test if we can also start from CO2 fixation itself to then produce formate and then go all the way to this activated methyl group. So what Giuseppe did is basically CO2 fixation in situ at 25 bar, what I did during my PhD as well with nickel iron powder as a catalyst and also a better electron donor in the form of iron. And after 16 hours, he would get also a very similar yield to just starting from formate. That means 13% of 5-methyl H4F. So basically what we showed here is that we can make a mineral and cofactor-assisted complete methyl branch just starting from CO2 and hydrogen, which was really exciting for us. So looking at other cofactors, I already mentioned redox cofactors a few times. There can be electron and hydride donors and acceptors, so there come in different shapes. As I said, there are flavins, such as flavin adenine nucleotide, also FMN, also F420. There are very many cofactors that look like that or similar to that. And of course, we also have the nicotinimates such as NAD. So just briefly, so I can also present to you the work that Oskari Lehtinen has done since he started his PhD roughly 1 year back. And Manavika, who just started her PhD now. And flavin reduction does work for us. We tested a lot of different minerals and conditions to actually see under which conditions we could abiotically reduce flavins, and it's still going on. So just as a snippet of the work, you can see here that we see a difference depending on what metals or minerals we're using. So nickel and iron work quite well under 2 different pHs. Iron sulfide still works a bit, but we cannot really make it work with the oxides that we used. But nevertheless, we know that we can abiotically reduce FAD to FADH2. That means we can also then use it as a abiotic electron donor in further reactions. So, but what I wanted to focus a little bit more on in the next 50 minutes roughly is nicotinamide reduction, because this is basically how our whole cofactor journey started, together with Frida Pereira, who was my first PhD student, now is my first postdoc. And basically what she did most of her master's and PhD is trying to get NAD produced, and not only NAD, but also precursor versions of it. So what we started with is we have here NAD+, and the first premise was, okay, can we reduce NAD+ to NADH? So what Delfina did is she used different iron nickel containing metals and mineral powders. Back then, like 4 years ago or 3 years ago, we used a quite high ratio to the cofactor, so a lot more metals than cofactor itself under certain conditions. We also always did controls under argon just to see what would happen if we wouldn't have any hydrogen in the mix. So what we then observed is basically just that you have seen the NMRs before, because this is the main analytical means that we used for these endeavors, is we see over time. So you have a timestamp here at half an hour, then you go to 1 hour, 2 hours, and 3 hours, and 1.5 hour, 1 hour, 2 hours, 3 hours. I'm not sure why that changed. I'm very sorry, but you can see that there is an increase in 1.4 NADH. This is all a little bit mixed up right now. I'm very sorry. And what we could see here is that we basically can first of all see that there's a structural difference between NAD+ and 1.4 NADH. And we can also see that we can use this peak to quantify what we get. And what you can also see just during the time, we can really monitor how much NADH we get. And the question is, we also are getting this one biologically relevant version of NADH and not a reduction on any of the other sides. We get a little bit of 1.6 NADH as well, but we later found out that it is mostly basically acidically catalyzed reaction starting from 1.4 NADH. So, and what we observed throughout time is that iron and nickel promote the reduction in very different ways. And what you can see here is we did different reactions back then. So we pre-treated the iron nickel powders before we actually put any buffer with reaction on it, so dryly. So we pre-treated on hydrogen. And we, of course, then also did control reactions under argon. And we did the same for nickel. So what you can see here, if you pre-treat the iron with hydrogen before the reaction is quite fast. But what you can also see is that for iron, it doesn't make a huge difference when it comes to the reaction if it has hydrogen during the reaction or not, because iron is a great electron donor by itself. For nickel, it looks a little bit different. So back then, we really needed to pre-treat it on the hydrogen so it really works. And it really does not work if you only have argon in the mix, because nickel's redox potential is not enough to actually reduce NADH or basically produce hydrides. What we then wanted to know is the mechanisms that were at stake. So we conducted experiments in heavy water just to see if there's a difference. So if we could actually see if iron could split up the hydrogen in the atmosphere to give us NADH, or if iron could also use the protons, basically produce nascent hydrides on the surface to also then reduce NADH. And we saw that iron indeed could do both. So what is happening on the surface? As I said, iron can be an electron donor, both an electron donor as also a catalyst for the hydrogenation, while nickel is mostly a hydrogenation catalyst for this reaction. After that, there was Delfina's first paper in her PhD. We wanted to get a little bit more natural in the conditions. So we looked at nickel-iron compounds that you would also find not only in surprising systems, but also, for example, meteorites, and also decrease the metal NAD ratio 1:1, and also decrease the concentration of phosphate buffer that we actually had. So this was all in collaboration with Harun Tüzes, whose lab basically helped us producing, or they made these nickel-iron compounds. And you also can see a trend now, which was really interesting for us to see how, for example, adding nickel to iron can increase the yield of 1.4 NADH quite highly. So the interplay between nickel and iron was really important to increase the yield. If you only have nickel and iron, for example, you would not get as much, but it would only work under argon if you had still enough iron around to work as a electron donor. So this was all quite nice to look at, but then Delfina had a really great idea to understand the chemistry even a bit better, because we went a little bit more natural in the conditions, but of course NAD is quite a complex molecule already. So Delfina thought, well, if you look at a lot of different organic cofactors here, they all share an adenosine-derived tail, and the question is, do we even need that tail, because the actual chemistry is happening here, here, here, and here, and here. So what is actually the role of this tail? Could it have does it only have an enzymatic role? So basically that enzymes can actually bind to these cofactors, or as it was already stated a few decades back, or is there even more to this tail, at least when it comes to NAD? So what Delfina started doing is doing reactions with nicotinamide mononucleotide, so basically NAD without the AMP. So, and when we started these reactions, it was really surprising to us that there was quite a big difference between the two, because when you start, so we used first of all the mineral powder that worked best for NAD. So we started here as at 1 hour. Sorry, and after hour. So first of all, after 1 hour, you would get a lot of 1.4 NMNH, so the one that actually gets reduced here. But then throughout time, the concentration of 1.4 NMNH would decrease, and you would get a lot of side products. Then we got really interested into like, okay, there seems to be a structural difference between the two. So NAD really seems to shield these overreduction reactions. So first of all, we want to see what is actually happening. So what are all the side products that we are getting? So we got some help from the NMR department at the University in Marburg in the form of Shulan, who helped a lot, and also Nicole from the MPI in Marburg, who helped us with liquid chromatography to figure out what we're actually looking at in these very complex NMRs. So what is actually happening? So we get 1.4 NMNH in our reactions. These yields are what we get after 4 hours, but we also get a lot of side products. So there is even a side product that is fully reduced, that is basically there from the very beginning. But the rest is basically side products starting from 1.4 NMNH. So you have another reduction reaction, but you also have a hydrolysis reaction here that goes to this hydrolyzed form of 1.4 NMNH. So it's really interesting for us to see. Just that you can also see it in a time frame. So you can see that with the same mineral powder, 1.4 NADH is slowly increasing and basically is quite stable, but it's also not as quickly reduced as NMNH. So NMN is being reduced to NMNH quite quickly, but then the one single reduced form of it is going down over time, while the overreduced forms and hydrolysis products are going up over time. So it's not a very, in comparison to NAD, not a very stable hydride donor and acceptor. So we also wanted to know where this variety comes from. So we also looked a little bit more closer, what does each metal do, because we started with nickel-iron compounds, and we also figured out that nickel is actually the one that increases the side products with the mononucleotide, while it actually, when you remember, once you put a little bit of nickel into iron, it increases the 1.4 NADH yield. So there's a big difference there. So what the take-home message for us was here is that the environmental conditions, like for example, what type of metal you have around, could also steer the selection of cofactors, in the sense of, if you look at cofactors in an abiotic environment actually doing pre-enzymatic reactions, if you had a nickel deprived condition, NMN might have been a good hydride acceptor and donor, while when you had a lot of nickel, NAD might have been the better one. Just as a thought. I'm not saying it is what it is, but it might have been a good influence for this. So why is why is there even a difference between the two? So the one thing that they have not in common is the dinucleotide structure of NAD. And it seems like this structure enables a more specific reduction, but how? So what we know from literature, there was just a great paper last year that NAD actually comes in a folded formation. So it actually folds in an aqueous solution. And the question is this actually also shielding from side reactions? It seems like it is, because this is the main difference that both of them have. So we also conducted direct competition reduction experiments, and there we saw that 1.4 NADH is at least 30% higher than the 1.4 NMNH, independent of the catalyst that we use. So if we use nickel, it's still more NADH than it is NMNH. So really in a mock competition experiment, NADH always comes out on top. So, and for that, so we now know when it comes to the reduction of these cofactors, there's a big difference. The question is now, how does this even translate into the back reaction? Because it's nice to actually reduce NAD plus and NMN to NMNH and NADH, but we also, in a prebiotic environment or en route to an alkylated network, we would also need NADH and NMNH to be reducting agents to actually give away their hydrides again. So first of all, we conducted cyclic voltammetry just to see if there's a difference between the reaction products in NMN reduction and the 1.4 NMNH itself. And you can, as you can see, just very briefly, you have here the Redox potential of NMNH, of one single reduced NMNH, and then the reaction product, whatever is in the reaction or in the solution of the reaction, comes a little bit later. So it would not be as good as a reductant. So that means the side products of NMN reduction are worse reductants than 1.4 NMNH. That's observation one. And the second observation was thanks to a great paper by Joseph Moran and Robert Meyer from 2024, because they showed that NADH can be used as a reductant with the help of several metal ions. We tried it with iron 3, so we used their conditions to see if NMNH and NADH can both reduce in this proxy reaction pyruvate to lactate. It's not about what reaction we're looking at, but just rather, is it even possible to reduce anything with NADH and NMNH? And what we saw is that the yields that we get from NADH and NMNH are comparable. So they are comparably good hydride donors, at least under our conditions. So last but not least, this really means, what would this mean in a prebiotic scenario? So we have NAD and NMN just as an example that we hope can also transfer into other cofactors, that under certain environmental conditions, you would get a very specific reduction product, for example, from NADH, but you wouldn't get as much from the NMN one. So you would have a selection process between the two, just again as a proxy. Just imagine a lot of other molecules here as well that might be in competition with these molecules. So, and then you would get a detachment of basically a hydride from the surface with the help of these cofactors, and then you could get to different conditions, for example, to reduce in solution oxidized organics with reduced organics. At the same time, you would have a lot of side reactions with certain cofactors, or cofactors to be whatever you want to call it. So that means the more stable a certain molecule was, it could have had a major influence on what would happen in a prebiotic scenario. So I hope that kind of made sense. It's a little bit hard also to talk to only my screen because I cannot see anything. But anyways, I hope I kind of conveyed the message that it is worth, and it is really worth looking at the reactions of cofactors in a non-enzymatic scenario, and also look at how their structures are actually giving some prebiotic function as well. So as a last slide, I just wanted to sum up what what I showed so far. So for us, it is possible to connect organic cofactors to mineral-assisted CO2 fixation, which is really great to see. We also postulate that there could be a geochemical selection of crucial biomolecules that we should consider and also look into more. And the main question that we're still working on is if it's possible to create a non-enzymatic reaction network based on cofactors starting from CO2 and hydrogen. And for that in the future, we also want to see if cofactors can also interact with each other, for example, reduce certain steps along their own biosynthesis, et cetera, et cetera. And with this, I would like to thank you, and I'm ready for questions.
Percy, thank you for your presentation. It was really good. I was wondering something, but it might be very specific actually.
Yeah.
As we know, Luca is believed to have had an RNA-based immune system. What might be the connection with the, did you hear?
Yeah, it's a Moody paper you're referring to, right? Yeah.
Yes. What might be the connection with the viruses in the early stages of life?
Oh, that is a very specific question. That is probably not the best one for me. I honestly don't know. So I think if you read the paper, they have very good insights on that, how that could actually refer to any like why an immune system could have been important or not. For me, that is something very advanced already. I mean, you see what we are doing, and it's quite very simplified, right? So I think at the stage where we need an immune system, that is something that I usually don't think about that much, because I think we are in the very early stages of like a chemical selection, so to speak, on the route to Luca. Nevertheless, that I think what that shows, and also what your question shows, is that how advanced Luca was most likely already. So that what the prebody chemistry that we're doing to reach Luca is very, very far away from what Luca already was. Luca is basically the last, when we're saying last universal common ancestor, it's the last thing we can reconstruct with genetics, right? So it's what I think it shows, and I think also what Moody and co-authors say in the paper is that there was already an environment that most likely Luca had to kind of protect itself from. So that means there were probably also already other communities, etc. So that's all something we have to consider in our work as well.
Okay, thank you so much.
And I have a question too, but we read a few articles about Luca with our team members before and discussed about on it, but one of these said that kind of like this. Can you check from the chat part and I can read it? Luca aged back to the 4.9 to 4.33 billion years ago. And we also know that the Earth's age near the 4.55 million years. But by this time, we know that the Luca's age is 3.6 to 4.2 billion years. So can we say that the age gap between Earth's and Luca's age shorter than we know?
So yes, I think, I mean, that's at least what the paper said, right? We actually discussed that. So we had like a journal club in our basically whole institute, because there's a lot of bioinformaticians there that, I mean, I'm not, right? So I had to rely on what people tell me there, or what their insights are on that. So although this is a very interesting thought, for me that wouldn't change too much on how I would look at Luca, to be honest. And also from what I understood, and I hope I'm not stepping on any toes, is like that these molecular clocks are not that 100%. I think I know Holly Betts, for example, I know they're doing amazing work. So the thing is, I'm not sure if I would really put a lot of inferring on based on the actual age, because the problem that we have is, like, for example, if you do any phylogenetics, right? So of, I don't know, dinosaurs or whatever, the one thing that you actually need is a fossil to actually date it. And we don't really have that in a way. So we have to rely on molecular clocks, but it also means that we have to be very careful on where we place LUCA, also when it comes to the geology, etc. So that's also why I always try to be, might not have been so clear, but I've tried to be quite agnostic about the whereabouts of LUCA, because, for example, water-rock interaction systems are quite universal. And I think they have been there for a very long time, as long as you had hot rock and water. So I think what I would take away from that is that we have to be a little bit more agnostic about where LUCA lived. And I think that Moody's paper also does it quite well. I mean, they said it could have been surface, it could have been deep sea. We have to consider a little bit more than that. And I think that's also encoded into the time issue. So when was it exactly? But I think the broader we are a little bit and more open, the more we can look, if that helps. Again, I'm not criticizing molecular clocks. So just what I understood and what colleagues told me, I think we should not be like, okay, this is exactly how it was, because I think we don't know enough.
Thank you for answering and taking your time. Sayın Doktor Martina Preiner'e konuşması için teşekkür ediyoruz. Kendileri adına Tema Vakfı'na fidan bağışında bulunduk ve bağış sertifikasını e-mail üzerinden ileteceğiz.
We extend our sincere gratitude to Dr. Martina Preiner for her presentation. We made a donation of saplings to the Turkish Foundation for Combating Erosion, Reforestation and the Protection of Natural Habitats on their behalf. We will send the certificate via email.
Thank you.
Değerli misafirlerimiz, şimdi Sayın Doktor Kaan Çandru'yu özel ayrıcalıklı biyoloji yoktur, astrobiyolojide yaşamın kökenleri olarak bir tesadüf modeli konulu sunumunu yapmak üzere davet ediyoruz.
Dear guests, may I now invite Dr. Kwon Chandru to present his talk on No Privilege Biology, the Origins of Life as a Contingency Model in Astrobiology.
So, can I start now?
Yes, you can.
Hello, everybody. Thank you for having me today. Today, I'll be talking a model that we published last year in regards to how we deal with non-biomolecular origins of life or astrobiology concepts. So the title is basically No Privilege Biology, the Origins of Life as a Contingency Model in Astrobiology. I know it's a bit loaded, but I'll get to what I mean by the title actually. Just before I start, I would like to have some disclaimers actually. Our group basically focus on non-biomolecular or agnostic as a spelling error, it's not astrological, it's astrobiological framework, which means that we are not overtly concerned about how essential prebiotic biomolecules, for example, RNA, amino acid, metabolites, are formed in prebiotic setting, or how they assemble or contribute to the emergence of primitive life, nor whether they are used in the search for ET life as well. We have been working on this non-biomolecular prebiotic chemistry concept since a little bit earlier than 2017, and we needed a framework to place all our experiments and also our thoughts within the astrobiology context. This paper and presentation place our work and thoughts within that framework. So as an introduction, this is the paper which we wrote last year. It's called Alternative Pathway in Astrobiology, Reviewing and Synthesizing Contingency and Non-Biomolecular Origins of Life. And it's based on several concepts. First, which I'll go through specifically. First is the N equals to 1 problem, the messy prebiotic chemistry, the non-biomolecular origins of life, and macroevolution, which we kind of make it into how chemical evolution of life on Earth and elsewhere could have happened actually. So before I continue, I want to introduce some jargon keys. The first one is macroevolution, which means that evolutionary processes over a very long time scale. The second concept and the third concept are related. One of it is, it's philosophical to a certain extent. It's called determinism. In biological sense, determinism is expressed as convergence, meaning that the repeatable evolutionary outcome is determined by natural selection, leaving no room for any other alternative possibility, therefore making outcomes predictable. So when you look at the word determinism or convergence, when I use, it means that it is predictable. So it's like a dominoes, a row of dominoes. When you push one, everything follows a certain pattern. On the contrary, contingency is a concept which is cost dependent, meaning a sequence of prior causative event governs a particular evolutionary outcome, making the outcome unpredictable. Like rolling a dice, you will not get the same outcome again and again. So in terms in biology, convergence, which is the evolutionary determinism, organisms or life has a limited way to adapt to certain functionality. Here are two pictures generated by AI. So there's some weird stuff as well going on, but I can still tell you what's the concept. We somehow only have one way to swim, which is using wings, even by previous reptiles, ichthyosaur, dolphins, penguins, different sort of life. But when they swim, they use their fins. And also we have only one way to fly, which is using wings. That means birds, bats, and bees and insects uses regardless of the evolutionary lineage. Therefore, what this is suggesting is, again, evolutionary outcomes are predictable. And it's supposed to be very ubiquitous across nature. Even with the idea of convergence, if at all we assume that dinosaurs were not extinct, it would likely that mammals will not rise, meaning that a humanoid body plan would have still emerged and prevailed. So Russell and Seguin question how the selection of larger brain size could result in anatomical changes in a Velociraptor kind dinosaur. So basically what they did is by taking a Velociraptor and increasing its brain size and making its anatomy to match, to sustain this big head, it turns out that it will come out very similar to humanoid. It's also been named as dinosauroids. So this is an example of how convergence will happen regardless whatever changes happen actually. But the big question is, if evolution is predictable, then extraterrestrial life or life elsewhere might also exhibit similar characteristics. So this is the idea actually. Even if you find life elsewhere, it's likely going to be similar to life on Earth. So if you find microbial life elsewhere, it likely have the same, membranes and probably DNA and metabolites as well. Okay. On the contrary, this is about contingency actually. As I mentioned earlier, it's a dependence on evolutionary outcome on initial condition and prior events. It suggests that if these initial conditions or events were different, the outcome would have been altered. So kangaroos are very, very unique mammals. They are not similar to mammals elsewhere. They're only found in Australia. They jump, they hop, they bear their babies without placenta. They're very different actually. And similarly, a platypus, which is a duck-billed, web-feet, furry body, further demonstrate that this organism is very, very different. When the sample of platypus was brought from the New World to the Old World, the taxonomists at the time thought it was a fake animal being stuffed together with different parts actually. Interestingly, the platypus is also one of the few mammals among its class that actually lay eggs. So the idea here is that this makes evolutionary outcome very unpredictable and highlight the role of chance in evolution, because considering that Australia was separated from the main land at that time actually. And it also contests that convergence might not be universally applicable as commonly thought as well. This makes, again, evolutionary outcome very unpredictable and highlight the role in evolution. So this idea of evolutionary contingency is basically coming from this book, from this biologist called Stephen Jay Gould in a book titled A Wonderful Life, The Brugus Shale and the Nature of History. The Brugus Shale is this fossil bed in British Columbia, if I'm not mistaken, in Canada, where they featured unique soft body animals in very weird features actually. And a chance event happened that the Cambrian extinction event wiped out most of them about 500 million years ago. So this is how they look like in their fossilized way, and this is the artist's depiction of it. So here is Hallucigenia. You can see on your left here actually, and this is the artist description of it. And you can see they're not sure whether it's walking this way or that way, although the artist here just picked the way and walked. And here is Opobinia, which is also very a weird creature as well. It has 5 eyes with this kind of a claw-like thing snapping on this thing actually. So this is to show that we don't see this kind of animals anymore actually. This is all wiped out completely. So Stephen Jay Gould, in his famous quote, said that any replay of the tape of life would lead evolution down a pathway radically different from the road actually taken actually. He posits that some of the survivors of the Cambrian extinction, who wiped out most of these weird-looking creatures, became the fauna that we are seeing now actually. So without that Cambrian extinction event, we will not see body plans like what we are seeing right now on Earth actually. So to put this in a pictorial sense, actually, I'll go through one by one. This is the idea of contingency, which you have many parts leading to different outcome here. And this is convergence, which there are many parts, but leading to a single outcome actually. And there are a mix of these parts, combination parts where contingency plus convergence. The key word here is that contingency means unpredictable, different outcome. Convergence means predictable and similar outcome. So what's the context here with astrobiology? In my opinion, astrobiology is largely deterministic actually. The origins of life involve the formation and organization of biomolecules to build components and working system necessary for life, which is in other words chemical evolution. We use them as biosignatures to detect life elsewhere as well. And we usually are given a framework like this. So if I start from abiotic chemical space, which produce a lot of chemicals, and then within some minerals and whatnot and everything, and then you have some kind of chemistry thing, reduction most of the time with basic compounds here like hydrogen gas, nitrogen gas, methane, hydrogen cyanide, and then it forms your amino acid. Your amino acid polymerized to become your protoprotein, and then it forms secondary or tertiary structures to make protoenzyme, which can do ancient metabolism to become a proto-life. The same thing with basic chemistry here as well. You can form your nucleobases, your nucleotide polymerized to become your oligonucleotide to become a proto-RNA, and proto-ribosome capable of self-replication and to become your proto-life. So this whole part down here is your prebiotic chemistry, taking a step-by-step way to become your proto-life, which some people can call it as a last universal common ancestor as well. So if we transpose this slide, again, I use this arbitrary term throughout the paper as well, non-life to life and life-likeness. And here, similarly, abiotic chemical space, which has a lot of chemicals. So they provide the basic chemicals that become chemical states, which are all this circle here. The blue is driven by biomolecules and the white are non-biomolecules. And I'll tell you why we have non-biomolecules as well. So the blue one goes on to become life and become LUCA and become life as we know it. And the white one, because we don't see it now, become dead end actually here. So the lines and arrows here are showing chemical transformation of chemical states. The further lighter they go across the board here, it becomes more lifelike. In this system, we only assume that there's only one way to make life, which is deterministic, as I mentioned before. And of course, there are many other deterministic parts where may arrive at LUCA, which we don't see, but this is how it's depicted. But this representation is kind of problematic actually, because we have this n equals to 1 problem. Statistically, having only one sample to say how things work is kind of poor. And we humans are generally very, how to say, we are human, we are prone to make mistakes because of our observer selection bias. There's a lot of bias that and this is one of it, because we see life as this all in everywhere on Earth, so we assume life elsewhere, and the origins of life also would likely use this kind of biomolecules as well. So that is that. And most origins of life are biocentric, ignoring this a key point here, where the realistic scenario of prebiotic chemistry, which is essentially messy. I'll show you how is this, why we call it messy actually. So a lot of these messy prebiotic chemical spaces are found in all prebiotic contexts. You can find them in hydrothermal wells, Miller-Urey experiments, carbonaceous chondrite will arrive on Earth, which are pristine prebiotic / abiotic chemical spaces, are dominated by non-biological chemicals, with only a small, very tiny fraction of them are composed of biomolecules. So this is how the assumption works actually. So on your right here, or your left, I think, this is life as we know it, which we know is made by protein, DNA, RNA, fatty acid, carbohydrates, lipid, metabolites, sugars, and all this on. They are made, largely to, sustain information, have boundaries, perform metabolism. So all of this, we assume, this is biology as we know it, is coming from prebiotic chemistry, which is basically producing a lot of junk material, a myriad of organic compounds. Some call it junk, some call it tar, some call it garaguta. So almost all prebiotic chemistry will produce all these messy chemicals, a lot of combinatorial chemical stuff and everything. And only a very select few of them will become, make your amino acid, RNA, and stuff like that. So to demonstrate how complex this is, what I mean by prebiotic chemistry is messy, is before 2009, when the Murchison meteorite fall happened, and using just normal analytical chemistry, and using one by one analytical chemistry, we could only detect about 600 molecules. And this paper, which I like a lot, is published in 2009 So they use a non-targeted method to see how much of compounds you can find in about a few grams of this carbonaceous chondroid. So carbonaceous chondroid are interesting because they represent pristine condition formed during the formation of the solar system. So whatever chemistry you find inside there would likely happen on early Earth as well. And there are similarities with this as well. So this technique they use, which is FTICRMS, they found that there are about almost 14,000 unique compounds. And if you include structural and stereoisomerism, you only get like almost like 10 to the power of 7 compound, which is quite a lot actually in that sense. So with these many organic compounds, which are non-biological, with just a few milligrams of sample, it seems unlikely that scarce biomolecules in this messy prebiotic environment are sufficient and uniquely reactive to preferentially form biomacromolecules and functionality selectively actually. This is the conundrum we have actually. In every prebiotic settings, we have a lot of non-biomolecular chemistry going on, and we have very little biomolecules going on. And there is no magic to happen to just select only these biomolecules to become life as it is actually. We knew this issue quite long then, and in 2014, we started to ask ourselves the question, what if we ignore this reductionist view of biology, meaning that if you break down all the small components, the 20 amino acids, the 4 nucleobases, the 2 types of lipids being used, and just start with prebiotic chemistry, what would happen if you apply just energy and put it under geochemical condition? What chemistry will rise and emerge? We want to see the process more than the actors of it actually. So the answer we thought at the time, and of course we're not the only one thinking about this as well. Eddie Pross also has thought about this as well. The answers is basically chemicals that are opportunistic will do stuff. You know, you want to just see the chemistry. We don't care where it goes. In other words, thermodynamics might be the first criterion. So we then tried to do some experiments with non-biomolecular prebiotic chemicals, which we choose here. To prove a point, we choose hydroxy acid. So they're basically congeners of amino acid. The difference is just they have a hydroxy group here against an amine group here actually. So we selected 5 of them to see whether they can form polymers under standard prebiotic chemistry. We also know that in theory that thermodynamically forming ester bonds, which hydroxy acid can do, is much more easier than what amino acid can do to form polyesters actually. There's a reason why amino acid is kind of selected, because this amine group enhances hydrogen bonding in protein secondary structures, and also peptides are generally less prone to hydrolysis. Because you can form easily polyesters using hydroxy acid, you can also lose them via hydrolysis as well. Nevertheless, we did the experiment. We took 5 different kind of hydroxy acids, which I have shown here. Actually, the name really doesn't really matter. And we wet dry them, just a single wet dry cycle for 80 degrees. The initial experiment was done for 1 day. And then if you do it for longer, you remove a lot of water from it, and you found a lot of copolyester gels. Copolyester, basically, they are combinations of different kind of hydroxy acid forming polyesters, which form a gel-like state actually. And when we take this gel-like state and we add some surfactant in this case acetonitrile, they phase transition to become micro droplets. And these micro droplets are showing, we call it membraneless micro droplets because they don't have a membrane. They form via liquid-liquid phase transition, and which is also ubiquitous in biology. And where they can able to hold some dyes, some RNA dyes, they do compartmentalization of these dyes within them actually. So we published the paper. All these are great. This is just a model of our non-biomolecular prebiotic chemistry. It shows you can form polymer, you can make big macromolecular structures forming compartment-like, prebiotic compartment-like entities. There are other types as well of this kind of concepts. It's PNA, XNA, dapsipeptide. So another example is dapsipeptide. This was done by Nick Hart's group, where dapsipeptide are basically a polymer or oligomer made of amide and ester bonds. They have both these bonds together. They seamlessly polymerize under wet drying cycle. So this group, they did many times of wet drying cycle using lactic acid and also alanine, and they realized that each time they do the wet dry cycle, the enrichment of alanine, which is an amino acid, becomes more and more. Actually, this is what the data is saying. So in other words, the alpha hydroxy acid here seem to aid the addition of alanine in the dipeptide. In other words, scaffolding the formation of prebiotic peptides actually. So this is what the aid of non-biomolecular chemistry helping the formation of more biomolecular chemistry. So then our next question is, I showed you the concept of contingency convergence. I showed you the concept of non-biomolecular origins of life. So what happens when you plug in the non-biomolecular origins of idea to contingency actually? So, okay. So based on this diagram, which you can read in the paper in more details, we just want to show two things actually. First, we think that the origins of life on Earth is likely to be contingent and may have been scaffolded by non-biomolecules. So we can show you here that the white circles, which are non-biomolecular, could have aided these biomolecules, chemical states to become life as we know it. There are some end states here as well. The dead ends are also here, and they could have scaffolded the formation of life as we know it actually. So the lines here show that the white non-biomolecular chemical entities are influencing the blue biomolecular chemical state to be selected to become LUCA. This is in complete difference with the one which I showed you earlier actually, where you assume all biomolecules will work together and they'll form life as we know it, and none of the non-biomolecules which exist in abundance will just die off actually. So this is what we are talking about actually. The idea here is to show that these non-biomolecular white circles can be anything. Alpha hydroxy acids is just a model, but they could be a lot of other chemicals out there which could have scaffolded the formation of life as we know it. And this is the earlier thing showing that deterministically how this is done. So the second thing which I want to talk about is that origins of life elsewhere other than Earth could also be contingent actually. Is that in these white circles, again represented by non-biomolecules, could have led on to become life as we don't know it. Different from what we know of LUCA, that might have developed on other planetary bodies. The scaffold, in other words, the scaffolding can work in two directions too actually. So what I'm trying to say here is that because we have been somehow selected contingently to have this 20 amino acid, 4 nucleobases, and so on, by our environmental factors and our geochemical system across like 1 billion to 500 million years ago. These other chemicals which we don't see in life on Earth could have also played a role in making up life as we don't know it actually. So I showed you two distinctions. One is the deterministic point of view and the contingency point of view. One is origins of life on Earth, which shows that these non-biomolecular chemical states could have influences this biomolecular chemical state and then wiped out because we don't see it. There's no fossilized life at the time. And also, if we were going to detect life as we don't know it in other planets, it's likely to be played by non-biochemicals as well. But these are two binary concepts. But we know the examples which are shown, life is not that simple, which have two concepts and you can see which one wins. They are likely to be a spectrum between contingency and determinism, would likely to exist. So here you can see, again, contingency could have played earlier on in the beginning part of this life-likeness, which is more closer to abiotic chemicals, and later on become more and more deterministic actually. This is one way we can look at it. The second way is the reverse. Determinism pushes biochemistry to become life at ease, and towards the end, right before becoming life as we know it, contingency could have played a way actually. So you can see the spectrum between this, from contingency to determinism, from determinism to contingency. Likely also a mix between contingency and determinism would also likely exist, meaning that there's a point where determinism exists earlier on during the life formation, contingency happens in the middle, and then it becomes deterministic again. This can happen in the opposite direction as well, contingency, determinism, contingency as well. So this is essentially what I'm trying to say. And we have some open question notes and caveats with this model. The model was created to formally incorporate and evaluate our hypothesis about non-biological origins of life experiment, fitting this idea into explainable framework actually. We somehow would need replication no matter what, and it could be an RNA, DNA, XNA, compositional or other unknown system for life elsewhere. The model does not specify the constraints necessary for conditions that lead to the emergence of life. This is done deliberately to make it simple to understand and grasp as well. And this represents only one prebiotic chemical space. There are many prebiotical chemical space which exist. Even arrival of carbonaceous chondroid via panspermia delivery as well could change the chemical space in that way, could lead to different contingent ways as well. The model also is devoid of a higher level of abstraction, such as bootstrapping to some form of biology, if there is such a thing, or when such bootstrapping happens as well. So finally, my take-home message is that as for, there is no specific reason to believe that life as we know it is made prebiotically by biomolecules during the origins of life, especially since prebiotic chemistry is messy. Other molecules may have scaffolded the origins of life to life as we know it, and there is no specific reason for or against the belief that extraterrestrial life would conceive of the building blocks similar to modern biology. We are interested in this concept of an alternative explanation for the origins of life and using them to find life elsewhere as biosignatures as well. I'd like to thank the core people which we have worked with and we are talking quite often as well. This is a huge group of people that we are always engaging in. And I'd like to thank a lot of our funders and sponsors before as well. That's the link to the paper. Thank you very much. I'm ready for question and answer.
Sayın Doktor Kuan Chandra konuşması için teşekkür ediyoruz. Kendileri adına Tema Vakfı'na fidan bağışında bulunduk ve bağış sertifikasını e-mail üzerinden ileteceğiz. Şimdi 10 dakikalık soru bölümüne geçiyoruz.
We extend our sincere gratitude to Dr. Kuan Chandra for his presentation. We made a donation of saplings to the Turkish Foundation for Combating Erosion, Reforestation and the Protection of Natural Habitats on their behalf. We will send the certificate via email. We now have 10 minutes for questions and we encourage our participants to ask any they may have.
We have one question in chat box. As you mentioned, there are many hypotheses for the origins of life, one of which is the Theo-Star world. How can we explain the sources of energy and the organization of processes in this hypothesis?
Can you repeat that again?
Okay, as you mentioned, there are many hypotheses for the origins of life, one of which is the Theo-Star world. How can we explain the sources of energy and the organization of processes in this hypothesis?
What is a TL star world?
TL star world.
I sent it to chat from here too, if you want to check the question. Yeah.
Oh, Theo-Ester world.
Yes.
Okay. Theo-Ester is interesting compound actually. We have done some work with it. We have some ideas about it. It's just that how you maintain Theo-Ester in a watery condition, which is a problem because the hydrolysis really fast. So you want to reduce the water activity. I think that could be that actually. To form Theo-Esters, I think there are many ways, but to maintain them in watery condition is the problem. Probably it could be having once the first cellular unit is already formed, a Theo-Ester could probably be maintained inside there as well. That's my idea, I guess.
And we have a question to another about cyanosulfidic prebiotic synthesis. Do you think cyanosulfidic prebiotic synthesis could also be relevant for extraterrestrial environments such as Enceladus or Titan? Again, I can see them here.
Is in the chat box?
Yeah, chat box.
In the YouTube chat box, I assume?
No, it's here. Google Meet. I sent it.
Oh, okay. Let me see.
I think there is another scenario for the prebiotic.
Okay. Do you think that cyanosulfic prebiotic synthesis could also be relevant for extraterrestrial? Yes, of course. I think like any chemicals can be applicable as long as it has enough concentration or enough energy to make it do stuff or reactivity.
Okay. So thanks for answering our questions. I think that's all.
Thank you so much.
Değerli konuklarımız, bir sonraki oturum için 10 dakika ara verilmiştir. Son oturumumuz saat 3'te başlayacaktır. Oturuma katıldığınızı belirtmek için chat kısmında ve aralarda paylaştığımız linklerden katılım formuna ulaşabilirsiniz.
Dear guests, a 10-minute break has been given before the next session. Our last session will begin at 3 p.m. To confirm your attendance, you can access the participation form via links shared in the chat and during the breaks.
Değerli misafirlerimiz, şimdi Sayın Dr. Eva Stuyken'i erken yaşama sınırları konu sunumunu yapmak üzere davet ediyoruz.
Dear guests, may I now invite Dr. Eva Stuyken to present her presentation on limits of early life.
Okay, thank you very much for the invitation to this meeting. So I'm happy to share some of my work and my thoughts on what may have limited life on the early Earth and what we can learn from that for the habitability of other planets out there. So there's been, over the last several years, several studies that have proposed that perhaps primary productivity on the early Earth, so about more than 2.5 to 3 billion years ago, may have been much, much lower than it is today. So some people argue, as you see in this figure as one example, that perhaps primary productivity was about 1,000 times lower than it is today back in the Archean. And so what can we learn from that for our search for life on other planets? That's my question. And so before I get into that, before I get into more details, I want to show you a really kind of simplified view of how life really works, kind of from a perspective of a geochemist. So fundamentally, so the green circle here is a cell. So imagine this is a microbial cell. And so fundamentally, all life on Earth, as we know it, requires some kind of electron acceptor and an electron donor. And these two are then reacted with each other in the cell, and they provide energy. And there are waste products coming out of this. And then this energy is used to create new biomass. So there's always a carbon source, which is either organic carbon for heterotrophs or CO2 in autotrophs. And there are other nutrients, nitrogen, phosphorus, and various metals that are needed, that are then pulled into the cell with this energy and converted into new biomass. And so if you want to think about the limits of life, we need to really think about these four general types of ingredients: electron acceptors, electron donors, the carbon source, and all these other nutrients, which in themselves each can be limiting. So as an example, to explain this a bit better, so think about chemoheterotrophs, which are, for example, include us as well. So our electron acceptor is oxygen. Our electron donor is organic carbon. That's the food that we eat. And so from eating this food and breathing oxygen, we gain energy, and this energy is then used to make new biomass. In our case, also the carbon source is organic carbon, and the other nutrients are also organic bound, nitrogen, phosphorus, and metals, and so on. So that's how our metabolism would fit into this. And so in our case, all of these ingredients can become limiting at some stage, potentially. But so we are not really relevant in the search for life elsewhere. So we need to look at autotrophic metabolisms, the metabolisms of primary producers in this scheme. So let's first look at chemoautotrophic anaerobes. These are organisms that, for example, could have existed on the early Earth. And so a chemoautotroph means, chemotroph means this organism is taking two chemicals. In this case, in this example, I have sulfate here as the electron acceptor and hydrogen as the electron donor. Can you hear me?
Yes, we can, but we cannot see if you change your slides. Sorry for that. Yeah, thank you so much.
Should I start over?
No, that's not. We can continue in here.
Okay, so as an example, the chemoautotrophic anaerobes, they would use sulfate as the electron acceptor and hydrogen as the electron donor, as in this example. So these two chemicals are reacted with each other, they generate energy, and this energy is then used to make biomass. And so in this case, because an autotroph, CO2 is the carbon source, which is then pulled in, and it's using various dissolved other nutrients as nitrogen, phosphorus, and metal source. So there's one metabolism that's relevant to potentially early Earth and other planets. And in this case, all of these ingredients could potentially become limiting. CO2, maybe not so much, because we tend to have a lot of CO2 in the atmosphere on the early Earth, certainly. But certainly all the other three types of ingredients could become limiting for this metabolism. Another important metabolism to think about is oxygenic photosynthesis. So that's the most important metabolism of primary producers on the modern Earth. And so what's really key about this metabolism is that the electron acceptor is the photosystem that's been excited by the photons, and the electron donor is water. And so that's important because it means that these two ingredients, so the electron donor and the electron acceptor, are essentially both unlimited in their supply. So these organisms have an unlimited supply of energy. So they need CO2 because they're autotrophs, but CO2 also tends to be fairly available. And so as a consequence, really the most limiting ingredient for these types of organisms tend to be the nutrients, nitrogen, phosphorus, and metals. So these organisms that dominate primary production on the modern Earth are really completely unlimited by energy. They are really only limited by nutrients, and that makes them so powerful. They're also anoxygenic phototrophs. I want to mention those for completeness. So anoxygenic phototrophs also have a photosystem that acts as the electron acceptor. But they need another electron donor, which is not water. So this is an example here out of photoferrotrophs, which would use Fe2+ as the electron donor. And this can become limiting. So these organisms, they can become iron limited, in addition to becoming potentially nutrient limited. Okay, and then before I move on, so there's also another concept I wanted to mention is energy hierarchy. So organisms that have photosystems, they tend to get a lot of energy out of those. So they have a lot of energy to use, which also makes them quite powerful, or makes this metabolism very powerful. And then the second most powerful energy supply is oxygen. So that's why animals like us, we can we have so much energy available to us because we have this powerful energy source of oxygen, of aerobic respiration. Whereas in contrast, organisms that use so chemoautotrophs that would use CO2 as their electron acceptor, like methanogens, they actually don't have that much energy available to them. So there's another sort of hierarchy in energy supply that also factors into this discussion. Okay, so what does it mean now for astrobiology? So it means that before oxygenic photosynthesis evolved on Earth, productivity was really constrained by any kind of planetary processes that supplied nutrients and energy. So remember today, we have primary productivity is completely dominated by oxygenic phototrophs. So almost all primary producers are oxygenic phototrophs in the ocean and on land. And so they're independent from energy, but before they appeared, energy may have been limiting. And so therefore, we need to look at these kind of planetary processes that supply not just the nutrients, but also the energy. So once oxygenic photosynthesis evolved, it liberated the biosphere from energy constraints, and it then also became only nutrient limited. But now, of course, the problem is that oxygenic photosynthesis evolved only once on Earth. So there's only, as far as we know, it was invented only once. And so we have no guarantee whatsoever that this metabolism would also evolve on other planets. It might, it's possible certainly, but we need to also consider the possibility that other planets may have a biosphere, but they may not have oxygenic photosynthesis because that metabolism was just maybe never invented. And so what we then should think about are planetary processes operating on those planets that supply not just the nutrients, but also the energy for a pre-photosynthetic biosphere. And then think about how those planetary processes would then constrain habitability on those planets. So to figure that out, so I'm a geochemist. I work on the rock record of the early Earth and try to learn from that rock record what that tells us about the habitability of other worlds. And so now the question is, if we now look at Earth rock record, what can we learn about these planetary energy and nutrient supplies from the rock record before oxygenic photosynthesis became dominant? And so before I delve into that, I want to show this graph, which is the compilation of carbon isotope data through Earth history. So it's starting about 3.7 billion years ago. That's the first data point here. Then all the way up to the modern. And shows in green organic carbon, organic biomass that's contained in rocks. And in blue is carbonate, so limestones and other things like that, other carbonate rocks. And so the key point is that this difference between the isotopes is really a reflection of autotrophic metabolism. So it tells us that since about 3.7 billion years ago, life has existed on this planet and taken CO2 into its cell, like you see in the schematic here on the right side, and converted that into biomass by some kind of autotrophic metabolism. So we know that autotrophy has existed and primary production has existed on Earth for a long time, almost basically as long as our rock record goes back. We can also learn from this that life has been microbial for almost all of Earth's history, and so therefore microbial ecosystems are the best analogs for an extraterrestrial biosphere, probably. But now the question is, so we know autotrophy existed. When did oxygenic photosynthesis emerge? And once we know that, what powered the biosphere before that event? So pre-photosynthetic biosphere, before oxygenic photosynthesis, what powered and limited that biosphere? Okay, so first question, when did oxygenic photosynthesis emerge? So we need to figure that out first. And so I'm going to show you two different data sets. One is the biological constraints. So this is the genomic record. So people have, biologists have essentially looked at the rates of evolution in microbial phyla, and using calibration points, worked out approximately when this phylum of cyanobacteria and the ability to produce oxygen first appeared. And so, so of course there are large error bars in these kind of analyses. But so what they came up with is that cyanobacteria, oxygenic photosynthetic cyanobacteria, emerged approximately between 3.0 and 2.7 billion years ago. So it's in the late Archean. And then they expanded further and diversified a bit more a bit later than that, so between 2.7 and 2.5 billion years ago. And then they continued to expand and evolve, of course, after that, up to the modern day. And so it means that roughly at this point, so when they first emerged, and maybe probably when they also diversified and expanded a bit more, that would have been the time then when, according to this analysis that I showed before, life would have been kind of liberated as a whole from its energy constraints. So from that point onwards, we would expect then life to be dominated by life to be mostly nutrient limited. And so now we have a time point. So before 3 billion years ago, we need to then look at the biosphere and think about these energy limitation, nutrient limitations, so before 3 billion years, according to this data set. There's another data set from the geochemical community. There are several other data sets. I'm just going to show this one. So this is, don't worry about the details. The point here is that people investigated iron-manganese ratios and molybdenum isotopes in a variety of iron formations around the world from the Archean. And so they find this fairly strong correlation between these two proxies back to about 2.9 billion years ago. And so this correlation is very difficult to explain in the absence of oxygen. So the most plausible explanation for this correlation is that there were manganese oxides forming back 2.9 billion years ago. And these manganese oxides were also getting buried in sediments. They're not just forming in the sea surface, but they're getting buried in sediments. And then molybdate ions absorbed onto these manganese oxides and were isotopically fractionated. So that's what's explaining this, the x-axis, the range on the x-axis. And so to make manganese oxides and to also bury them in sediments, you need to have an oxygenated water column on continental shelves. And so this study therefore argued that back to about 2.9 billion years ago, there must have been oxygen in the water column, at least in the shallow ocean, down to sediment water interface to explain this data set. And so this is totally consistent with the biological data that I showed before. So it's looked at these two approaches really are converging towards a similar date, about 2.9 to 3 billion years ago, for the origin of oxygenic photosynthesis. I want to highlight that this does not say anything about anoxygenic photosynthesis, so this is essentially still quite unconstrained. So we don't really know when anoxygenic phototrophs evolved. Then, of course, just for completeness, I want to mention that about 2.4, 2.3 billion years ago, that's when many proxies suggested oxygen wasn't just being kind of locally produced, but actually became quite abundant in the atmosphere and in the surface ocean. So I'm not going to go through these in detail, but each of these lines here that are listed is an independent piece of evidence for an oxygenated surface ocean and oxygenated atmosphere. There are banded iron formations that disappear, pyrite and uraninite that disappear from the rock record, so they're oxygen sensitive. Red beds, they are continental iron oxides, essentially very, very shallow water iron oxides that start to appear abundantly. Soils, the chemistry of soils changes from being iron depleted to being iron rich, iron oxide rich in particular, across this time. And then there's this record of mass independence of isotope fractionation, which tells us about atmospheric chemistry. There are several indicators that all say that about 2.4, 2.3 billion years ago, the environment became more pervasively oxygenated. And so the picture that's kind of emerging from this is that is shown here. So according to these studies, then oxygenic photosynthesis appeared around 3 billion years ago, then maybe diversified, and oxygen kind of slowly accumulated and eventually oxygenated the surface of the Earth and the atmosphere at around 2.5, 2.4, 2.3 billion years ago, somewhere in this time window here. So that would be called the GOE, or the Great Oxidation Event. And so this bottom figure here shows oxygen in the ocean. So shallow waters would have been kind of transiently oxygenated in the Archean and then fully oxygenated from the Proterozoic onwards. But the deep ocean remained anoxic and iron-rich. That's important. I'll come back to that later. So, but now the key point from the slide really is that if we want to understand the pre-photosynthetic biosphere before oxygenic photosynthesis evolved and took over and became really dominant for productivity, we should really look at life before 3 billion years ago. So that's kind of our cutoff. That's our maybe most conservative cutoff at this point for the emergence of oxygenic photosynthesis. Okay, so now let's think about this. Let's go back to this initial cartoon that I showed. So what were the electron donors that were available before 3 billion years ago? And I think because some people were not able to see the slides, I'm just going to briefly cycle back to this. So in the beginning, I showed this cartoon, which is essentially a geochemist's view of biology. So the green circle is a cell, and the cell I mentioned needs electron donors and it needs electron acceptors to generate energy. It needs a carbon source and it needs the nutrients. And so now the question I'm asking is, what would have been before oxygenic photosynthesis? What would have been the electron donors and the electron acceptors? Okay, so going back to this. So what were the electron acceptors that were available before 3 billion years ago? So remember that oxygen would have been absent. So this is by default kind of ruled out. And so this is a busy slide, but so one important electron donor would have been supplied quite readily by volcanism. Would have been sulfate and elemental sulfur that ultimately came from SO2 gas. So we know that volcanoes outgas SO2. They do that today, and we have kind of indirect evidence that they were also doing that in the past. And that's based on the sulfur isotope proxy that I mentioned before. So it's a particular isotopic anomaly, capital delta 33S. Don't worry about the details, but the key point is that this anomaly, so on this graph you see these positive and some negative values. They can only be produced, or they can the largest values of these in this parameter can be produced if you have SO2 gas that's interacting with UV radiation in the atmosphere. There are a few other mechanisms, but that they can really not explain this pattern. And so really this data set is evidence for a variety of things, but among other things for this slide, the key point is that it's evidence that we had volcanic outgassing of CO2, of SO2. And this SO2, we know, would then have rained out into the ocean. Some of it converted to sulfate. So we have some direct evidence of that. So we have some barite and other sulfates preserved, also some elemental sulfur and sulfides. But so this sulfate and elemental sulfur would have been available as an electron acceptor. But now, so it's a very small source compared to today. So on the modern world, oxidative weathering is the major supply of sulfate. And so in comparison to that, it's still a relatively small source. And at the same time, there would have been a lot of reductants available. And so people have calculated, based on kind of isotope mass balance modeling, that probably the reservoir of sulfate in the ocean would have been less than about 10 micromole per liter. Compared to 28 millimole today. So we're talking about 3 orders of magnitude less sulfate in the Archean Ocean compared to the modern ocean. So there would have been a limited supply for chemoautotropic sulfate reducers. The next one that's interesting to look at are nitrogen oxides. And these also can be supplied by kind of indirectly by volcanism. So if you have volcanic outgassing of a lot of CO2, and that CO2 is then hit by lightning in the atmosphere, along with N2 gas is contained in the atmosphere. Then what happens is that in that reaction, the CO2 molecules and the N2 molecules are completely shattered and broken apart. Then as the lightning stops, these atoms essentially recombine and make new molecules such as nitrogen oxides. And so on a completely abiotic world, there would have been a constant supply of these and potentially concentrations of up to maybe about 1 micromole per liter. That's a sort of abiotic scenario, up to 1 micromole per liter of these nitrogen oxides. In comparison, the modern ocean contains 30 micromoles per liter of nitrate. So it is. So we've also replicated this experiment in the lab, and so also in our experiments, we have shown that it is true that under an Archean atmosphere, this plot here on the right side, under an Archean atmosphere with lots of CO2, we do produce a lot of these nitrogen oxides, but then in this model that's shown on the left side. They then calculated how much of these nitrogen oxides actually accumulate in water if you have a lot of iron, dissolved iron around. And so they found that you get maybe up to about 1 micromole per liter of these nitrogen oxides. So it's a very, it would have been a constant supply, or sort of, I guess, erratic, but you know, over long term would have been a constant supply, but again at a small reservoir. And so the next one, probably the most abundant electron acceptor on the early Earth, would have been really CO2. That would also have come from volcanic outgassing. So this plot here on the left shows estimates of CO2 in the atmosphere through Earth history. So there are a variety of studies that have tried to estimate CO2 from different proxies, and then this thick black line and the yellow bar that goes through that is based on models as well. And so we know that probably more CO2 was needed to keep the Earth warm because of the faint young sun. People have estimated based on these variety of proxies and models that perhaps CO2 was on the order of 0.01 to 0.1 bar to maintain a warm climate, or climate. That's in comparison to only 400 ppm today. So CO2 was definitely much more abundant in the atmosphere on the early Earth than it is today, and it's supplied by volcanic outgassing. And so CO2 would have been an electron acceptor, would have been available as for energy. But remember from this other plot that I showed in the beginning, it's actually a very poor electron acceptor. So it does work, and there are organisms, there are methanogens that happily use it, but it doesn't provide a lot of energy. So it's a relatively low energy yield compared to, for example, the energy that you would get from oxygen or nitrate or something like that. Okay, now I want to transition to the major nutrients, nitrogen and phosphorus, and also minor nutrients, metals that were available before 3 billion years. So remember, we need electron acceptors, electron donors, and nutrients as our key ingredients in addition to carbon. So now on to nutrients. I also already talked a little bit about nitrogen. So nitrogen today in the modern biosphere is almost entirely over 98% or so, derived from biological N2 fixation. So these are enzymes, microbes that have enzymes that can take N2 out of the atmosphere and convert it into ammonium. But the enzyme probably hasn't always existed. So now, again, phylogenetic reconstructions where they try to reconstruct the antiquity of these enzymes, and they have it going back to about 3.2, 2.9 billion years ago, intriguingly, same time as oxygenic photosynthesis, which may not be completely coincidence. It's possible that perhaps there were sort of other enzymes doing a similar job perhaps before that. We don't know. We have no record of them really. That's quite speculative. So, but it is also entirely possible that this enzyme did not exist before 3.2 billion years ago. And so we need some other mechanism to supply nitrogen as a nutrient to the biosphere. And so again, lightning would have been one of those. So lightning can supply nitrate, as I just showed you. And also hydrothermal vents can supply that. So I'm going to discuss these a bit more in detail. So this is just another data set. There's geochemical evidence for nitrogen fixation starting about 3 billion years ago, 3.2 billion years ago. So we have geochemical and biological evidence that nitrogen fixation emerged or existed by about 3.2 billion years ago, but it may have been absent before that. And so as I said, lightning could have been one of these sources of nitrogen, bioavailable nitrogen on the early Earth. So you saw this plot before, actually. So these are some of our experiments, but other people have also done experiments like this. And so in these experiments, we find that if you have an Archean atmosphere with high CO2 and also maybe a bit of hydrogen, so you produce fairly high amounts of these nitrogen oxides, not too different actually from the modern atmosphere. So here on the right side, you see the nitrogen oxides that we produce in a modern atmosphere. And the order of magnitude is actually similar, within an order of magnitude, or within a factor of 2 or 3 or something like that. The composition of the oxides is a little bit different. That's what these red and blue dots show. But over time, if these oxides all dissolve in water, they would have been available as some sort of electron acceptor. The Proterozoic may have been actually the time of less nitrogen oxide production. But importantly, on the early Earth, when we had high CO2 levels in the atmosphere, those would have been then enough to generate high levels of nitrogen oxides potentially. Regarding phosphorus, the other major nutrient, that's extremely controversial right now. So geochemists, essentially like me, are useless for reconstructing phosphorus in the early ocean. So if you look at some of the papers that have been published about estimating phosphate levels in the Achaean, they diverge by 5 orders of magnitude. So they cannot agree. And so we need to really do better from the geochemistry side. So right now, phosphorus is really poorly constrained. There is perhaps some help here from the biology again. So this is a data set where so one of my postdocs, she led this, looked at enzymes that uptake phosphorus from the environment. And what she found is that the oldest enzymes that emerged first are actually those enzymes that are adapted to high phosphorus concentrations, high phosphates. And those enzymes that are adapted to relatively low phosphate concentrations actually emerged fairly late in Earth's history, comparatively late. And so the conclusion from the study is that maybe phosphate actually declined over time, over Earth's history. And so therefore, then it would mean that maybe phosphate was actually quite abundantly available on the early Earth. Okay, so now this means that we maybe had a fairly rich source or moderately rich source of nitrogen, bioavailable nitrogen from lightning and also from hydrothermal vents. I didn't put that in here, but so hydrothermal vents can also supply nitrogen abiotically as a form of ammonium. And we also, apparently, according to at least these biological estimates, we may also have had a moderately large reservoir of phosphate on the early Earth. We don't really quite know it exactly how the phosphorus cycle operated. As I said, the geochemistry is fairly in disagreement about that. But potentially phosphate would have been made available by just enhanced weathering under fairly acid conditions. And so maybe we'll be looking at a moderately large phosphate reservoir on the early Earth. And then lastly, regarding metals, these are essentially the micronutrients that are important and essential for life today. So metals would have been probably moderately abundantly been supplied by hydrothermal vents. This graph here shows kind of a simple calculation of just fluxes. The black bars are is the modern river flux of, in this case, manganese, cobalt, nickel, copper, and zinc. The gray bar is the modern hydrothermal flux of these elements. And then the orange bar is the Achaean river flux. That's assuming same continental size, which is probably an overestimate. And the red bar is the kind of calculated, estimated Achaean hydrothermal flux. So it's based on thermodynamic modeling, and then paired with kind of just a water flux estimate. So the thermodynamic modeling is assuming these hydrothermal fluids come out, they mix with an Achaean seawater. Then we looked at how much of these metals are still in solution after mineral precipitation has stopped. And so then we take that number and plug it into just a water flux calculation. And so what you can see from this is that, so of course the modern river flux is the highest in most cases, just because of fairly high, oxidative weathering. As we have plants, we have a lot of stuff happening on land today. But if we look at the Achaean, on the Achaean Earth, hydrothermal vents would have been, certainly for zinc and copper, would have been much more important sources of these metals. If continents were much smaller than they are today, then perhaps also for nickel, hydrothermal vents would have been more important. And what actually was not considered in these calculations is that in some cases also maybe organic ligands may have helped and stabilized these metals in solution coming out of hydrothermal vents. And so really the conclusion is that's what's shown here on the right side in these graphs for copper and also for other metals. Probably hydrothermal vents were by far the dominant source of these metals to the ocean. And if you consider the possibility of organic ligands, then perhaps the reservoir of these metals may have been on the order of 1% or several percent, similar to the modern, so not as low as some people have previously thought. Really thanks to hydrothermal vents and possibly organic ligands that were stabilizing these metals in solution. Okay, so the implication now for astrobiology is that, first of all, oxygenic photosynthesis would have marked really a singularity in the evolution of life on our planet that liberated the biosphere from energy limitations. So the origin of oxygenic photosynthesis would have increased the nutrient demand. And so maybe that's actually the reason why, if I go back to the slide about nitrogen, maybe that's the reason why nitrogen fixation emerged about 3.2, 3 billion years ago. Perhaps it's because oxygenic photosynthesis appeared and had a much higher demand for nitrogen at this point. And therefore, it was kind of favorable to invent this metabolism as well. Regarding phosphorus, so perhaps the origin of oxygenic photosynthesis is also the reason why the phosphate reservoir goes down. So we have these high phosphate levels perhaps in the early Archean, and maybe the reason why phosphate then drops in the Mesoarchean, according to these data, is because oxygenic photosynthesis emerged and the demand for phosphorus was suddenly higher because life now was not energy limited anymore, it was only nutrient limited, and therefore had a much higher demand for nutrients. That may explain these trends. But now the important point for this talk is that the pre-photosynthetic biosphere, so before 3 billion years ago, would have been probably limited by electron donors, so especially for the methanogens, so put in hydrogen and Fe2+, and also by electron acceptors, so for sulfur and nitrogen metabolism. So it's really these energy sources that would have been the most limiting for the pre-photosynthetic biosphere. And so what I find really quite intriguing is if you go through these slides, let me go back, and I think while I had some issues here with the talk, so I skipped over some slides because of the issues with the full screen. I skipped over a few slides about the electron donors. So the major electron donors, iron, would have been supplied really abundantly by hydrothermal vents. So iron is supplied, so we have banded iron formations on the Archean Earth, and there's trace limit evidence that iron would have been the major source, hydrothermal vents would have been the major source of this iron. The iron reservoir in the deep ocean may have been on the order of 0.1 to 1 millimole, according to these thermodynamic models that you see here on the right side. So in this particular paper, they calculated iron concentrations in equilibrium with various minerals between pH 7 and pH 8. But now more recently, people have suggested that the ocean was perhaps actually slightly more acidic, so maybe 6.5. And so we are probably looking more at the upper end here of this estimate. So on the sort of millimolar range. So iron would have been quite abundant. Yeah, we know from trace elements that the flux of hydrothermal vents was very high. So we know that from europium anomalies in banded iron formations, that there was a much stronger hydrothermal activity in the Archean Ocean than there is today. So this proxies europium anomalies that you see on the y-axis. They are produced at high temperature and high temperature fluids. And you see this anomaly is high in the Archean. There's actually a peak in the Neo-Archean that still needs to be explained. It's quite low today. And so the interpretation is that there was just a lot more hydrothermal volcanic activity on the early Earth. Then hydrogen, the other important electron donor, especially for methanogens, would have been overwhelmingly produced by hydrothermal activity. So this paper here on the right side summarizes various sources of hydrogen gas on Earth today or anytime. And really the most abundant one would have been hydrothermal alteration. So it's basically serpentinization, where water interacts with mafic, ultramafic rocks, minerals, and produces hydrogen. And so estimates are that hydrogen would have been up to maybe possibly 0.1 bar, and coming out of mostly hydrothermal vents. So really, oh yeah, this is another slide that I added in to show that on the early Earth we had a much hotter mantle, probably. That's evidenced by various proxies, and also so one that I like here is shown in the top right, the abundance of komatiites through time. So komatiite is a rock type that really only forms if you have fairly high temperature upper mantle melting. And so it's I think the most obvious evidence for kind of a hotter mantle, and therefore probably more extensive magmatism and hydrothermal activity. And so I think that's probably the most important takeaway message, is that, going back to my conclusions, that it was really volcanic processes, volcanic hydrothermal processes, plus or minus atmospheric reactions, such as UV photolysis for the SO2 and lightning for the nitrate, that would have been the sources of these chemical disequilibria, of these electron donors and electron acceptors. So in other words, if we're looking for habitable worlds out there, we really need to look for planets that are volcanically active, as possible, and also have an ideally atmosphere that also contributes to chemical disequilibrium by generating things like sulfate and nitrate and stuff like that. So that's really my key conclusion. So we should look for these types of planets in the future. And with that, I'd be happy to take any questions.
Sayın Doktor İva Öztürk'ün konuşmaları için teşekkür ediyoruz. Kendileri adına Tema Vakfı'na fidan bağışında bulunduk ve bağış sertifikasını e-mail üzerinden ileteceğiz. Şimdi 10 dakikalık soru bölümüne geçiyoruz.
We extend our sincere gratitude to Dr. Eva Öztürk for her presentation. We made a donation of saplings to the Turkish Foundation for combating erosion, reforestation, and the protection of natural habitats on their behalf. We will send the certificate via email. We now have 10 minutes for questions, and we encourage our participants to ask any they may have.
There is a question. It's also thought that hydrothermal vents are a possible point for the beginning of life. Are there any biosignature studies conducted in these areas? What are the difficulties of working in these areas? As I know, you have articles about biosignatures, right?
Yeah, so there's certainly biosignatures going back to the earliest rock records that are from areas that are hydrothermally active. So, for example, 3.5 billion years ago, that's when probably most people would agree that we have evidence of life on Earth. And actually, a lot of that evidence comes from a site that was probably some kind of volcanic caldera with a sort of volcanic lagoon, potentially. That's the one model that's been proposed. With active hydrothermal activity. And so, yeah, and also many other sites that are younger or similar age often involve input of hydrothermal nutrients or energy sources. So I think so it's difficult to quantify that based on these biosignatures, but certainly we certainly do see that life was interacting with these kind of hydrothermal fluxes. We cannot easily say from these data how dependent it was on those fluxes. That would be the next step to then kind of I guess look at those data sets more quantitatively. But certainly there is a spatial and temporal overlap. Yeah.
Thank you.
We have another question from the chat box, the broadcast. According to the theory of panspermia, it is believed that life on Earth originated from space. If life begins on other planets, can it be said that it can originate from Earth due to studies in space?
So panspermia, I don't know. I'm not a fan of the panspermia theory because I think it's just outsourcing the problem. So if we can make life on an asteroid, then why can we not make life on Earth? We have a lot more possibilities on Earth, I think. It's a much better place to be. Yeah, I'm not sure what to say. I guess there's this idea that maybe life could have originated on Mars and maybe was brought to Earth. So I think one of the main arguments for that is that like Mars was drier. So for a lot of these origin of life reactions, you often need to dehydrate organic molecules. So you need to, for example, if you want to make a protein, you can react two amino acids together, but you actually need to remove the water. And so some people have said that that's easier to do if you're in a place like Mars, where there's just much less water to begin with. There's more evaporation. So I could see that. I guess we just don't know really enough about the early Earth, but also about the origin of life to rule out that it could also have just happened on Earth. But if it did happen on Mars, I guess Mars was also hydrothermally and volcanically active at the time, and so then I think some of the same arguments would also apply there. So that probably hydrothermal activity and volcanism would also have been important to then start life on Mars if that's what happened.
Okay, thank you so much.
And I have a question too. When we have participated in a NASA competition, our aim was to create a chemosynthetic world. And I would like to ask a question that we were wondering while taking part in this competition. Do you think that life could begin in the presence of a chemosynthetic environment without photosynthesis? And how sustainably would this continue? And also, how complex could a life form be without oxygen gas?
Yeah, I think definitely life must have started without photosynthesis. So yeah, according to the geochemical data and also the biological data, at least oxygenic photosynthesis only started maybe 3 billion years ago. And so before that, there would have been at least 500 million years or maybe 1 billion years of life without oxygenic photosynthesis. So now the problem is we don't know when anoxygenic phototrophs emerged. They may have been older. We don't really have a date on those. But I think most people agree that it's a fairly complicated metabolism, and it's easier. There are some easier metabolisms to do that do not require photosynthesis. So I think most people agree that probably the first metabolism that appeared, first autotrophic metabolism would have been chemosynthesis. So then I guess the next question, how long could you sustain a biosphere with chemosynthesis? I think it really comes down to these sources of energy and nutrients that I talked about. So the problem of chemosynthesis is that you need to constantly supply the energy. So you need to constantly supply the electron donors and the electron acceptors. Whereas if you have photosynthesis, then you don't need to worry about the energy. So when you have chemosynthesis, you need to have a constant resupply of the energy, and that's then you have to think about a setting, for example, volcanically active setting, or things like lightning or UV radiation that supply that kind of energy. But that's possible. So probably on Earth it was happening for several hundred million years, probably. And then the last question was about how complex this biosphere could become without oxygen. So I think, yeah, there's probably a limit to how much energy you can get from this. Actually, some of the nitrogen oxides that I mentioned from lightning, they actually provide quite a lot of energy. Not quite as much, I think, as oxygen, but it's not far away from that. But the thing is, right now they're not very, very abundant. So you need to have a place or scenario somehow where you make a lot of these nitrogen oxides at a very high rate, so maybe extreme lightning or something, or some other process. And then that would maybe give you enough of this energy without oxygen. But it's it's kind of speculative at this point. But you would need to have another mechanism that supplies this energy.
Thank you very much.
And again, thanks for your presentation and attendance our conference.
Okay, yeah, thank you for the invitation.
Yıldız Teknik Üniversitesi Biyoteknoloji ve Genetik Kulübü olarak bu sene 4.'sünü düzenlediğimiz Astrobiyoloji ve Yaşamın Kökenleri Konferansı etkinliğimiz sona ermiştir. Bizleri onurlandırarak davetimizi kabul eden bütün konuşmacılarımıza ve siz değerli katılımcılarımıza teşekkür ederiz. Son oturumun katılım formunu birazdan paylaşacağımız linkten erişebilirsiniz. Gelecek etkinliklerimizden haberdar olmak için sosyal medya hesaplarımızı takip edebilirsiniz ve ayrıca birazdan oynayacağımız kağıt oyununa katılarak kitap ödülü kazanma şansını yakalayabilirsiniz. Sıradaki etkinliklerimizde görüşmek dileğiyle. Sağlıkla kalın, bilimle kalın.
Our Astrobiology and Origins of Life conference hosted by the Biotechnology and Genetics Club of Yıldız Technical University has reached its conclusion. We would like to thank all our dear speakers who honored us by accepting our invitations. And we would like to thank you, our esteemed participants. You may access the 5th session for your attendance certificate through the link to be shared shortly. You can follow our social media accounts to stay informed about our upcoming events. Also, by participating in the Kahoot game we will play shortly, you will have a chance to win a book prize. We hope to see you at our future events. Stay healthy, stay curious. Now we will give a short break of 2 minutes before moving on our Kahoot game.
Bye.
Bye. Herkese merhaba tekrardan. Kağıt oyunumuza ekranda gördüğünüz QR kodla ve pinle giriş yaparak katılabilirsiniz. 1-1,5 dakika katılımcılarımızın oyuna girmesini bekledikten sonra oyunumuza başlayacağız. ...51 geçe oyunumuza başlayabiliriz. Toplam 12 sorudan oluşuyor kağıt oyunumuz. Bazı sorularımız 2 katı puana sahip. Şimdiden yarışmacılarımıza bol şans diliyoruz. Kağıt oyunumuz sonucunda kazanan ilk 3 arkadaşımıza kitap hediyesi edilecek. 51 geçiyor. Oyunumuzu başlatıyorum.
İyi şanslar.
Çeşitli organik moleküllerin suyun içinde hidrofobik eğilimleri sebebiyle kümelenmeleriyle oluşan damlacıklara ne denir? Vesicle, mikrotübül, lizozom ve konservat. Burada gördüğünüz görselleri de yardımcı olması için koyduk biraz. Konservat diyenleri tebrik ediyorum ben kendi içimden. Bakalım kim demiş? Gotik. Tebrikler Gotik. Jüpiter'in hangi ayı dünya dışı yaşam için ana aday olarak kabul edilir? Benim gönlümden çok fazla Aquavert istemek geçiyor ama. Neyse. Europa diye. Ben dedim diyemedim. Gofrik ve gotik galiba.
Bir saniye. Cevap veremiyoruz. Bir soruyu görmeden süre bitiyor. Nasıl çözebiliriz?
Bir dakika o zaman. Tekrar bir 2 saniyenizi istiyorum.
Tamam. Yeniden yükle demeyi denediniz mi? Konuşamadım.